So Steve has received a PhD from Miami University in ecology, evolution, and environmental biology, and a master's in biology from the University of Texas at Tyler. His work focuses on applying genetic, ecological, and biogeographical inferences to guide endangered species conservation. It's a very different approach to Knauss' work. This is his PhD work, and the screen is yours, Steve. Thank you.
Yeah, so happy to share just a brief overview of some of the work that I did during my PhD. So let's dive right in. So we're going to be talking about freshwater mussels, so we'll get into those. So kind of a start here. I don't think it's any kind of shock to anyone that the current Anthropocene era is biodiversity is in major decline. And the loss of diversity itself is also, it's not equally spread amongst all taxa. And mollusks themselves, which I know most people probably don't even think about mollusks, are among the most threatened of species. So the group I'm going to be talking about today is specifically the unionid mussels. So these are freshwater mussels. And just to clarify, I'm not talking about the invasive zebra mussels or quagga mussels. These are native to the North America, and they're just ecologically, life history, they're very, very different. So just remember that. But you can see from these pictures that they come in all shapes and sizes, different colors. So they're a very diverse group of mussels. In fact, there's about 950 to 1,000 species, depending on who you ask at any given time. Unfortunately, about half of these species globally are listed as somewhere between near threatened or critically endangered and even extinct for quite a few. In North America itself is a biodiversity hotspot. We have about 300 species here in North America. However, unfortunately, the trends are very similar here, where already about 10% of our species are extinct. And about 65% of what remains is afforded some kind of protection at the local, state, or federal level. So some kind of endangered or threatened status and enhanced protections to come along with it. And so if you're wondering what the cause of this might be, it's really death by 1,000 cuts. There's all kinds of things that create this. So in the early 1800s, it was the button and pearl industry. Took tens of thousands of tons of mussels to make buttons for shirts. Agricultural, industrial pollution, kind of self-explanatory, runs into the waters, not great. Impoundments and dams, altering courses of rivers, invasive species. So you see on this native mussel, you have the congregation of zebra mussels that kill them, and then some invasive fish that eat them. Dewatering of rivers, obviously mussels need water to live, and declining fish hosts. I'll get why that matters in a sec. And also there's just a lot of unknown questions sometimes. We just don't know why they're gone in some places. So, and when I mentioned fish hosts a second, that's because the unionids have an extremely cool and unique life cycle. So as larvae, they're actually parasites on fish. So they attach, they kind of look like little tiny mussels, and they attach to the fish for a period of time, and then they drop off as very young mussels that go down into the sediment and grow up to be big mussels. So that's just the overview of mussels, because I know not everybody's familiar with them. So kind of my overall research goals with these mussels, but in general, most of my research really kind of focuses on applying kind of principles of population genetics, as well as some phylogenetics and biogeography to inform conservation strategy for imperiled species. And so, like I said, this is a portion of my dissertation work. So I have this broken up into three sections, three separate studies. So I'll kind of start going through those. So the first thing we would go through focuses on the entire genus of the entire genus is composed of 3 species. The genus is Platybasis. And in the entire genus is federally endangered, so I should also mention that all 3 species are. There is a lot of captive breeding and propagation efforts to kind of bolster the numbers in the wild of these species. The only problem is you can actually cause a lot of harm doing some of these propagation methodologies, especially when you're introducing young individuals into the wild by just near industrial scales. You can put so many mussels out, but you could have negative genetic implications if you don't consider it from the beginning and take appropriate measures. So the 3 species in the genus, there's P. caparianus, Platybasis caparianus. In this animal, there's actually less than 100 known individuals left. There's no genetic information for any of them, until this study. And I was able to sample the majority of these 100 animals for this study. The second animal is P. cipius, the sheep nose. So they're native to the Ohio, Tennessee, Mississippi drainage, and there are multiple populations known, and they're not quite as imperiled as Caparianus. Cicatricosis, also native to the eastern U.S. Unfortunately, there's only been 10 seen in the last 20 years, except for one that was morphologically ID'd, but not genetically ID'd, that I was able to include as part of this study. So again, here, this is just a sampling location, so the eastern U.S. And the way I actually sampled is with a buccal swab, so kind of like a mouth swab. You just kind of open the mussel a little bit and put a little swab in there. And it is a non-destructive way of sampling the DNA. And I should also mention that these mussels, this whole group, comes from very large rivers. They don't live in small little creeks, they live in very large rivers. And I'll get back to why that matters in a little bit. So just very brief overview of the methods. I sequenced them mitochondrial genes and used kind of a next generation method called RADseq, which really just the important part of that is it gives you a lot more data than some of the older methods. So here is kind of the major findings for Caparianus. So this was the animal that I said there's only about 100 known animals. But what you get is amongst the 2 rivers that it still occupies, there's only 1 population. So what that means is that they are able to interbreed somehow. So likely the fish hosts are able to move between those. And you see in the star-like phylogenies, so in the CO1 and ND1, what you get is these large, widely shared haplotypes with small satellite rare haplotypes. And what that really indicates is that there's actually genetic signal of population expansion, despite the fact that we know that they have dropped significantly. And what this is likely due to is kind of a lag time in genetics to keep up with current census population because they're long-lived animals. And overall, the effective population size comparative to many other species is actually very large. So that's what that NE value is showing. So that's actually a very large value compared to many other mussel species. So for P. cipius, which you see is actually 2 very distinct populations here. So within the Tennessee and Mississippi River. So that's what you're seeing when you see these 2 large peaks. It's basically showing that there is very little overlap in genetic makeup between the 2 populations. There's likely no migration between the 2. And another key takeaway here is that the Tennessee River is actually much larger of a population. So that's what's shown by the NE. So that's effective population size being in the 2000s versus 200. So it's nearly 10-fold larger. So when we look at that that one animal, the one that's only a couple have been seen in the past 20 years, and there was one found, but it was ID'd morphologically as that rare animal, actually turns out to just be one of the more common, the P. cipius species. And what that's likely due to is just extreme plasticity and morphology that you see with many mussel species. It's fairly common. It's unfortunate because it would have been nice to have found this extremely rare animal, but it turns out that it really wasn't. It was just I guess a slightly odd looking one of the more common species. So the overall takeaway is that we only have a single population of Parietas, but there are at least two distinct populations for Cipius. Both species actually have a very large population size for the most part, despite the fact that, well, genetic population size, and that single animal was not supported as being what it was ID'd morphologically as. So the conservation implications here is that the captive breeding strategies should be different between the two species, so there's not just a one glove fits all type strategy that can be used for the genus. So we really need to zoom in on species very specifically when we're kind of putting in place procedures to bolster the numbers. And also, there's a potential for undiscovered populations, so that gets back to this very large effective population size. And because these animals live in very deep, dark rivers, they're just very hard to sample for. So it's possible that there's reservoirs of genetic material out there that we just weren't able to find. So, all right. So moving on to the next study here. So this is a completely different species, still another freshwater mussel, still another unionid. It's in the Yosho mucket, and this species lives here in the central interior highlands, right? And so it lives kind of in this Four Corners region of Oklahoma, Arkansas, Missouri, and Kansas. Sorry, in that little area there. So the highlands themselves, they've served as glacial refugia. So as glaciers have expanded and retreated over the years, a lot of species have gone into retreat there, and many unique endemic species and populations have developed in that region. But we haven't really explored the mussel fauna all that much, despite the fact that there are quite a few species in there from a population level. So that's where that species lives. It also extends slightly to the west and into the lowlands. So you can see here, moving the, yeah. So this species is also, it's been reduced, 60% reduction in its size of its range. So it used to go out through this whole region, all these rivers. There are fossilized shells. So that's one very nice thing about mussels, is that the shells, they last for a long time. So we're able to easily characterize their historic distributions. But they're only found in about 40% of their previous range. And the little populations that do exist are very patchy. They're all cut off by dams because there's a lot of development in this area. So again, I swabbed the individuals. Here's the sampling locations for each of the animals and the sample sizes. Did many of the same genetic analysis that I did for the Plathobasis study. And what we see here is, again, two distinct populations. And this one is actually with a very strong border at the kind of the border of where the highlands, the central interior highlands stop and the central plains begin. So what you get is these two very distinct ecoregions with distinct genetic populations in each. And what you get is also that the plains is, again, a much smaller population than the highlands population. It's much less genetically diverse. And this is likely due to much more fluctuation in that region over the course of the last Pleistocene, or during the Pleistocene last glacial maxima, whereas the highlands population was able to stay much more stable. Oh, I think I just said quite a bit of this already. So there's two genetically unique populations. The plains is a smaller one, and they likely diverged during the last glacial maxima. And this is a fairly common thing that you see when you get large biogeographical barriers. And so the captive breeding strategies here, you really should not mix these two populations because there is likely local adaptation given that they've been separated for a significant period of time into distinct ecoregions. And also that the plains population itself being smaller is going to be more susceptible to further loss. So moving right along to this third study here. So here I used both neutral and adaptive loci to guide conservation. So really characterize population structure using adaptive in addition to neutral loci. So kind of one of the first steps when you're developing conservation strategy is to define conservation units themselves. So these are kind of clusters of populations that share traits within that unit that aren't exactly shared in other units. So for the most part, units should be managed separately from each other. And there's multiple ways to define them. Kind of historically, it's always been done using neutral genetic loci. So that's how I have done it with the other two species or the other two studies I talked about. But with this species, I was able to incorporate some of the adaptive methodologies. So it looks for adaptive loci as well. So the species itself is the federally endangered Poponitis popi. So it's a Texas horned shell. So it's native to the Rio Grande in the kind of Chihuahuan Desert region of the Southwest. There's only 5 known populations remaining. There was 4 until recently. We actually did quite a bit of surveying in northern Mexico and located this population. So if you aren't familiar with what that little piece sticking up in Texas is, that's the state of Coahuila. We're able to find another population down there. So there was some previous work that showed historical isolation amongst the Rio Grande Laredo population. So that's the most southerly versus the Black River, the most northerly. And this was found to be correlated with Pleistocene climate shifts and glaciation. But that study didn't cover those middle populations. So that's what I was looking to do here. And so just a quick overview of what the rivers look like. So they're quite a bit different amongst each of the rivers. They look very different. So they're influenced by different factors. So hydrologically, climatically. So again, similar genetic methods for part of it, but in this, as I mentioned, I was also able to incorporate some of the adaptive variation analysis. I was able to identify loci potentially under selection and reanalyze the population structure to see how that was basically defined by the adaptive loci in addition to the neutral loci. So this is similar to the other study. So this is the neutral loci here. We get these 5 genetically unique populations. So each of the rivers harbors a very unique population. There's not a lot of possible migration or gene flow or anything amongst the rivers here. But when we use some of the loci that are under selection, so these are the adaptive loci, you actually see a much tighter cluster of the two populations that are in the Rio Grande River, as well as the Rio San Diego, so that's that one in Coahuila. And what we found from this analysis was that the climatic and hydrological influences on these populations, which is likely driving a lot of the adaptive, a lot of the selection here, is very similar for those three. Whereas those other two populations, it's much different, and that's reflected here. So it is correlated with environmental factors. So takeaway, overall, there's 5 demographically distinct populations. They've all likely been isolated since the Pleistocene, and the selective pressures for those 3 populations at least are extremely similar. So when we're considering how to define some of these conservation units, that can very strongly guide interbreeding amongst populations if you need to do that, because you would only want to breed populations that have similar adaptive type loci. So overall summary, as I said, freshwater mussels are in strong decline. The principles of population genetics and biogeography can help to benefit the conservation efforts. Within each species, the distribution of diversity is also unique. So we can't just pick some kind of one glove fits all for different mussel species. They're all very unique. So unfortunately, that means that we have to study each one specifically before coming with a well-informed idea. And a lot of this data has actually already been applied to many of these species. So this was all done in high collaboration with different agencies working on these groups. So just some quick acknowledgements. There's a whole lot here, so not gonna have time to go through all of them. And with that, any questions?